Second thing is, second thing which is very, very interesting when we talk about the evolutionary aspect. Just now, I was just thinking, if I talk that E. coli completes its life in 20 minutes, and how many generations an E. coli can complete in a year. So I was just getting an idea, almost 25,000 generations, broadly 25,000 generations can be completed in 1 year. So when we talk about human being, if please correct me, we have so many evolutionary biologists. If I roughly say that human has been originated Homo sapiens somewhere 2 lakh years back. So till date, maximum 10 to 12,000 generations has been completed since the origin of human beings. So what bacteria can achieve just in 1 year, we have not achieved till date. So what I want to say, what is the level of evolution this bacteria would be having? Then, they, many times we call or when we teach that they're simple, but I always ask a question, are they really simple? Ultimately, what biotechnologists are doing, they are taking the tools from the bacteria and making some uses in the human welfare. We have taken the restriction enzyme, we have taken the thermostable enzymes, we have taken so many processes, and right now we boast of the gene editing, but the CRISPR has also come from the bacteria. So what I mean to say, when we talk about they are well equipped, so it's really a big question mark. Are they really simple when they are in such a small cell? They are performing all the basic functions of the life. And of course, when we talk about the gene transfer, when we talk about the higher organisms, normally we do talk about the vertical transfer of the genes. But in case of bacteria, they have both horizontal and vertical transfers of genes. So they're so advanced. They have very beautiful, they have developed during course of evolution, the what you call the defense mechanism, maybe in the form of biological warfare, maybe in the form of chemical warfare, maybe in the form of physical warfare. So they are really highly evolved, and they are evolving much more in a year than we have evolved since the origin. So anyway, probably we are going to hear much more on these scientific puzzles and aspects by our today's keynote speaker, Dr. Samir Giri. And thank you. My best wishes. And again, I congratulate the Zoology Department for hosting such a lovely international webinar. Thank you. Thank you, Aarti. Stage is yours now.
So much for your kind words. So like always, your encouraging words mean a lot to us. A pessimist complains about the wind, and an optimist tries to change it, and the leader adjusts the sail. The sails for this particular notion was adjusted by the person who is not only a great motivation to all of us, but also a visionary. He is none other than a respected principal sir, Professor Rajiv Agarwal. It would seem that due to some prior commitments, our respected principal sir was unable to join us for this webinar. However, he has sent us his blessings and well wishes for the success of this event. Now, I would like to call upon the President of Zoological Society, Soumya, to take over the event and introduce our honorable speaker of the day.
Thank you, Arathi, for giving me this great opportunity. Now, I would like to introduce our honorable speaker of the day, Dr. Samir Giri. Dr. Samir is currently the postdoctoral fellow, Genome Biology Unit at European Molecular Biology Laboratory, Germany. His primary research interests are directed towards gut microbiome, drug microbiota interaction, antimicrobial resistance, microbial community assembly, ecology and evolution of microbiology interaction, immanent properties of microbial systems, evolution of cooperation and competition. He received his PhD degree in microbial ecology and evolution in 2021 from Osnabrück University and Max Planck Institute for Chemical Ecology, Jena, Germany. He completed his bachelor's in life sciences in 2009 from SGTB Khalsa College, University of Delhi. He has received many fellowships and awards, including Eagleton Foundation Postdoctoral Fellowship, EMBL Symposia Fellowship, DFG Research Fellowship, and many more. His strong background in the research has allowed him to be a part of many editorial teams of various journals and funding bodies. In addition, he has also supervised many students. His research-oriented mind makes him different from others. He has also attended many public outreach programs. Also, he has numerous publications in renowned journals such as Nature Ecology and Evolution, ISME Communication, Current Biology, Current Opinion in Biotechnology, Journal of Molecular Biology, and so on. Last but not the least, he has very strong CV in his research field. Thank you, sir, for joining us today. We will have a pleasure being hearing you. I have an important announcement for all of you guys. All the participants are requested to drop down their queries in the chat box. The chat box will be made available for everyone after the session ends. The query section will be taken up by our second year student, that is Ajita Srivastava. Over to you, Ajita.
Hi, sir. I would now like to ask you to proceed with your talk and enlighten us all.
Sure. Can you see my screen?
Yes, sir.
Yes, sir.
Should I proceed or wait?
Yes, sir. Yes, sir. Please, sir. Please go ahead.
Yes. Cool. Yeah. So thanks everyone for different kind of introduction, but all good. And thanks Sandhya for the invitation, for giving me this opportunity to share the work that I was doing and currently doing for understanding how bacterial interactions actually shape microbial communities. So it's basically divided into two main parts. One is understanding how interactions affect each other, and also perturbations. The second part is about the perturbations that actually hamper those interactions. But then we can go one by one how to proceed with that. So we all know that bacteria are the unseen drivers of almost all ecosystems in our planet, and they are ubiquitously abundant and highly diverse. This you can also see from this image, that the number that 1 gram of soil contain like 6,000 to 50,000 species of bacteria, and the same in the gut microbiome. And these interactions are diverse, and only these interactions not happen in isolation, but they actually happen in communities. So most of the bacteria exist in a multi-species community, which you can see here is just an illustration to show that these are the swabs from the New York subway, like metros in New York, that when we swab these different regions from the metro, then and streak on the Petri plates that we grow the bacteria to see the colony forming units, that there are different type of bacteria that can actually grow on different media, and there are different colors and different size, different shapes of these bacteria. And within these communities, they are not only growing, they are also interacting, and these interactions are actually the key driving force of such communities. And when these interactions happen, they are not only happening within this, they are also driving various ecological or evolutionary processes. For instance, all of you might know that consortia of Nitrosomonas and Nitrospira actually do such processing of nitrification. So they reduce ammonia into different byproducts and then change into simple forms. In different insects, that bacteria that are residing in a special part of the body called bacteriome, they provide different nutrients such as vitamins and essential amino acid to the host cells that they are not able to produce. And the other aspect, that they also degrade complex molecules or pollutants, which they are not able to do when they are alone. So in a community, they can do much more better tasks rather than when they are alone. So it's like emergent property of a community to degrade toxins. But one of the most key important part of these interactions are, these are driven mainly by production and secretion of metabolites. And when these kind of interactions happens, these metabolites are secreted in the environment, provide new niches, and that niches open up new ways to grow bacteria utilizing the provided niches. So by this, bacteria not only grow alone, but also provide different environment to help other bacteria to grow. So broadly, if we talk about bacterial interactions in ecological or in evolutionary terms, there are two umbrella terms that we use that can be antagonistic interactions based on how two partners interacting each other and decreasing or increasing the fitness of each other. So in this case, one bacteria that is the arrow is indicating a Burkholderia bacteria is on the lawn of Bacillus subtilis bacteria. So when Burkholderia start growing, they start secreting some antibiotic that start making a halo. So, and this leads to decrease in fitness of Bacillus subtilis. But contrastingly, there are also synergistic interactions where bacteria not only help the other partner, they also increase the growth of the partner. So in this case, Escherichia coli is secreting a siderophore. Siderophores are molecules that are secreted in the pool or in the environment to capture iron, and the bacteria that are not able to grow can start grow when E. coli start secreting that siderophores. So these kind of interactions are common, but today's talk I'm focusing more on synergistic interactions. And one type of synergistic interaction that I focus in my PhD is called metabolite cross-breeding interactions. And these cross-breeding interactions are happened either in unidirectional way, where one bacteria produce something that other bacteria can take up, or it can also occur in bidirectional manner. So when both bacteria produce something, that each of them can produce something for the other partner and then grow better compared to in isolations. But this does not happen in one or two ways, but there are also like multipartite interactions, which is also known in nature, but it's difficult to study in laboratory. So to understand these principles, how these metabolic interactions actually drive in these communities, we start first understanding in a simpler manner in using synthetic ecology. So if we look into type of metabolite these bacteria produce, there are a range of different molecules that are exchanged between bacteria, either produced by bacteria or secreted by bacteria. Produced by bacteria or received by the bacteria. So there are carbon sources, nitrogen sources, amino acids, and vitamins or nucleotides. All these kind of essential metabolites that are produced by the bacteria can be taken by some other coexisting bacteria or by the community members, or also they receive, the producer can also receive these bacteria, these metabolites. And these interactions are not only common in bacteria, but also bacteria, but also between different eukaryotic and higher systems. And one more thing that recent sequencing-based analysis start accumulating. So I'm showing you in the bottom part this interaction map, which is a meta-analysis of literature search, indicating that that these metabolite cross-breeding interactions are really common in nature, and the nodes in the outer periphery show the phylum, and in the inner part is the orders and different interactions showing that they are exchanging different type of metabolites. The thickness of these arrows indicate number of metabolites they can exchange. So overall, the key message is that these kind of metabolites are really exchanged between different group members or between different phyla. And the key message is that this is actually common in nature. But the key message that is missing in the literature is actually what factors determine their establishment at the first place of such metabolic interactions. So this is a key question that I asked in my PhD, that what factors actually help them to start this. So when I started, when I started my PhD, there was a model proposed by Pandey et al. In this opinion paper, where they propose that when a autonomous cells start growing, they start secreting some metabolites, and these metabolites can be taken up by the neighboring cell. And when they start taking these metabolites, they start losing genes. So a mutant can arise in the population that can lose the gene that they are getting from the partner, and then a unidirectional interaction can be established. And then when these interactions started, then the chances are that bacteria that are giving something can also receive something back from the auxotroph. So in this part, the second part, the auxotroph the auxotroph is a bacteria that are not able to grow independently but depend on the outside source. That means either provided from the outside or given by the partner. And this can also happen in not only unidirectional way, but these interaction can transition to a bidirectional cross-building. When both of the partners start exchanging, they both lose some gene, and then they start exchanging both of the metabolites they required for growth. And this happens just by secretion and uptake of the metabolite, but there is no cost associated for the production for their respective partner. But when selection pressure, natural selection act on these bacteria to increase and interact for longer time, these interactions can be converted to or transition to a cooperative interactions. That means when bacteria grow, they start secreting these metabolites to their respective partners and cause a cost for these production. And when there is a cost associated, evolutionary theory predict that non-cooperator can also increase in these populations. But yeah, we can talk about this systematically in the later part. So the key question that I asked when I started the PhD is what factor actually facilitate the unidirectional cross-building interaction in the first place? Next question I asked, what are the propensity that unidirectional interactions can be transferred or transition to a bidirectional cross-building? And the third, I asked what happens when a obligate cross-building interaction established? Do they actually expand the niche of the interacting partner? And lastly, when these cooperative interactions are established, how they are maintained in the long run? Because I mentioned that when during cooperation, they are also propensity to increase the chances of non-cooperators, and these non-cooperators don't pay the cost of interactions and then collapse the interactions. So let's go one by one. So to answer these questions, I synthetically engineered different bacteria, different cross-feeding systems. So I deleted different genes of amino acid biosynthetic pathways from wild-type bacteria. So if I delete a gene, then the bacteria become auxotrophic. And that means that this bacteria can only grow when you provide an outside source of amino acid or grow with the partner. So this become dependent on amino acid. So when you co-culture two bacteria, which is a producer and an auxotroph, and then the donor, so I say the producer as a donor, and then this donor gives something that the auxotroph can grow. So with this, the obligate cross-feeding, unidirectional cross-feeding exchange can be established. And in another way, when both partners are co-cultured together, that means the two auxotrophs are paired together in co-cultures, then the obligate bidirectional exchange can be established. So I used both of these systems, but I'll go one by one and I'll tell you when I used unidirectional interactions and when I used the bidirectional exchange. So the first question I ask is about the factor that actually facilitate these interactions. So for this, I collaborated with Leonardo, Silvio, Shraddha, and Gada during my PhD, and this work is already published, so you can also read in details, but I'm just give you a snapshot of ideas that are actually found out in this paper. So to answer the questions, I took a number of different species of bacteria. So there are 25 donors that I plotted here in a phylogenetic tree. This phylogenetic tree actually constructed based on 16S RNA gene. And what are in unbold are the donor, that the donors are the producer of amino acids. So they can grow in minimal environment without needing any outside source of amino acid. And two species of auxotroph that I use are Acinetobacter bailey and E. coli. These are auxotrophic for histidine and tryptophan. So in total, I have 25 donor species and two species of histidine and tryptophan. And these are in the gray are the outliers to make the phylogenetic tree. So these are all firmicutes. These are all archaea. So to test this, I pair them in co-culture. So these are a donor. So I sometimes replace this. So donors are prototrophic strains, and recipients are auxotrophic strains. So when I co-culture them, in a deep well plate, and then grow them also in monoculture and quantify the growth after 24 hours. And with this, I can see that what is the difference in growth when they grow together versus when they are alone. All of the experiments were done under sterile environment. So what I show you here in the y-axis is the relative recipient growth. That means the growth of auxotroph when they are together with the donor, and the x-axis is what is the effect on these recipients. So when I divide this, there are negative interactions. That means in 1.3% of the cases, auxotrophs, when they were growing with the prototrophs or donor, decrease in growth. 35% of the cases, the interaction were neutral. That means there is no effect of being part of co-culture. And in 63% of the cases, the interactions established. That means the donor can support the growth of auxotrophs. And that this dashed line at 1 is indicating when the auxotrophs were actually growing alone. So there is an advantage to being part of co-culture when the auxotrophs are versus when they are alone. So I wonder what actually explained this 63% of the growth when the auxotrophs are in co-culture. So I decided to test the amino acid profiles of the donors, because my interactions were basically exchange of amino acids between two bacteria. So what I did is I collected, I again grow these two strains, the donor and recipients. So donor belong to 25 different species, and recipients are 4 types from 2 species. So I condition to grow them alone without, so the donor without amino acids, so grow them till exponential phase and collect the supernatant. That means the supernatant that is without donor cells, and feed it the donor cell with the to the auxotrophic cells, and also quantify the number of metabolites or amino acids present in the supernatant. So this I did by LCMS-MS. So I quantify the mass by the supernatant by mass spectrometry and what type of metabolites they are there, and also quantify when the supernatant is fed to is monoculture of auxotroph, can they support growth of the auxotroph compared to when they are growing alone. This is done by plate reader assays. So what I'm showing you first is the amino acid production profile by mass spectrometry. So so in the y-axis you see the donor genotype. These are different genotypes that you probably see in the phylogenetic tree. They are just coded by names. And the x-axis is different amino acid produced by the donors. And these arrows are indicating these are the auxotroph that I use for the main experiments. So overall, the main idea is donor secretes different metabolites or different amino acids in different a different quantity, not only quantity, but identity of metabolites or the amino acids are different in these produced by these donors. And this makes the interactions happen in the first place. Then I ask, can these produce amino acid explain the growth of the recipient? So I did the experiment of the when the supernatant was fed to the auxotrophic cells with and without supernatant. So what I plotted here is the focal amino acid. That means the amino acid that is the oxotrophic partner required. This is either tryptophan or tyrosine, or the total amino acids, because I can see from the heat map that they are not only producing their respective amino acid, but also the other amino acids. What I observed is there is a positive association between the focal amino acid and total amino acid, meaning that the production of amino acid in the supernatant actually explain growth, indicating that not only the required amino acids are uptaken by the recipients, they are also taking up amino acids that are secreted by donors. So possibly they are saving the cost of not producing those amino acids and taking the benefit from the pool of the hexametabolome. So to answer further, what other factor can explain the growth of the auxotroph in co-cultures, there were two ideas that we thought that time is that when bacteria grow, they prefer certain specific resources and habitat, and when they grow, because of the overflow metabolism and the leakage, they secrete a lot of metabolites in the extracellular pool. And then the idea was that when there are similarity between bacteria, the metabolic pool or could be more similar than when there are two dissimilar types of bacteria. And this lead to hypothesize that that the type of metabolite bacteria produce can also explain the growth of the cross-breeding interactions. So we decided to develop idea about the matrices that can explain the cross-breeding interaction. So we come up with 3 different measures of cross-breeding. So I will explain systematically what I mean by this is that amino acid distances. So the calculation that so the values I quantify using the heat map in the previous slide can be calculated based on distances. There's just a mathematical formula called Euclidean distance. I can calculate this by seeing that how similar my donor strains are producing amino acids similar to the recipient versus different. So the idea is more similar they are more likely to produce similar amino acid profile versus when they are more dissimilar. So when I did this, I find out that there is a positive association between the normalized growth of the recipient versus when they are plotted against the amino acid distance, meaning that more dissimilar is the amino acid profile, better the recipient grow. And this holds true also for phylogenetic distance. This distance was again calculated based on 16S rRNA gene and between the donor and recipient, indicating that when donors are phylogenetically distant, the crossbreeding happen much more better compared to when they are similar. And lastly, with the help of Silvia Vashina, we calculated the genome-based metabolic distance in these donor and recipient, and this was done using metabolic network calculations. You can calculate that how 2 different donor and recipient metabolic profile look like versus when they are similar, and this can give us a matrices which can be plotted in the x-axis, which is called metabolic distance. It's again based on Euclidean distance or Jaccard distance, and then plotted against the growth that I quantify in the first graph. And in this part indicates that that all of these matrices can explain growth, and suggesting that crossbreeding actually increase better or increase more when they are interacting more with the dissimilar partners than the similar partners. So I now transition to a second part of my talk, where I ask when a unidirectional interaction is established, what happened then? So this interaction stop here, or is there any chances that that this interaction can transition to a bidirectional crossbreeding interaction? So I just ask, what is the propensity that these interactions can be exchanged? So what I did, so in the previous part, I was talking about what happened to the auxotrophs. But in this part, I was looking into the donor side, what happened to the donor when they are part of the co-culture. So in this part, I again collaborated with PhD and postdocs in the lab, and this work is already published. So you can also look into the detail. But to tell the key idea that the experiment was same, the only thing was quantification was little different. So what I plotted here is in the axis is the effect on donor, and the y-axis is the relative donor growth. So when the donors are growing with the auxotrophs, they are negatively affected by donor, which indicate that they have possibly some cost of producing some metabolites that the auxotrophs were taking, so they are not getting something back. In 35% of the cases, the interaction was neutral. That means when they are in part of co-culture, there is no harm to the donor or recipient. But in 40% of the cases, these interactions were beneficial, meaning that when they are part with in co-culture with the recipient, they are also getting something back, meaning the reciprocity likely emerge in 40% of the cases. So interestingly, in all of these cases, I studied only cases that I showed in the previous case, like 63% of the interactions. So out of 63%, 40% of the cases have bidirectional interactions. So if you have questions, you can also ask me later what do I mean by this. So to summarize part 1 and part 2, so first I showed that donor can support the growth of auxotrophs in co-culture. And then I showed that phylogenetic dissimilarity between phylogenetic dissimilarity and metabolite dissimilarity actually promote cross-feeding. And lastly, I showed the recipient can also benefit in co-culture. So to move to the third part of the talk, I asked what happened when a bidirectional interaction is established. Does this interaction actually increase the niche space of interacting partners? And the niche space, I mean that number of nutrient sources a given partner can grow on. So in this part, I collaborated with Leonardo and Neela in the group. And to answer this question, first I'll explain what do I mean by ecological niche. The niche is well explained in our ecological textbooks, but in a very precise way, it's just the environmental factors that influence growth, survival, and the reproduction of a genotype that actually grow in that. And Hutchinson actually classically modified or updated the niche theory and divided this niche into fundamental niche and the reliant niche. Fundamental niche is basically the abiotic property of the environment, and the reliant niche is the interaction between the organisms. So interaction is really important. So when there is no interactions, that means any genotype can explore full potential of their fundamental niche. But whenever the interactions start happening, so this scenario rarely happens when the interaction is not happening, because all the organisms in this planet interact in different ways. So when antagonistic interactions happens, the niche can be contracted. That means the realized niche could be the part of fundamental niche, but this is even smaller in this case. And this is always portrayed in ecological textbooks, that realized niche is always should be the part of fundamental niche, but this is not always the case, because the synergistic interactions can even expand the realized niche beyond the fundamental niche. So what do I mean by fundamental niche is that if you imagine this box is as the fundamental niche, and the overlap is the realized niche, the synergistic interaction can expand this niche much more than the antagonistic interactions. So to answer the question that whether or not obligate interaction can expand the synergistic interactions and expand the ecological niche, I used again the bidirectional cross-feeding interactions as a model systems. And in this case, I used 5 species of bacteria. And those were auxotrophic for 4 different amino acids. They were arginine, histidine, tryptophan, and leucine. And I pair them in co-cultures and also in monoculture with and without amino acids. So with amino acids, they are in gray shade, and without amino acids, they are in white. And I put these different treatments in biolog plates, and these biolog plates are containing 31 carbon sources. They are 31 carbon sources. Those are different carbon sources that bacteria can utilize, and they are available in triplicates in 196 well plate. So when you put them together and quantify the growth, so whenever the bacteria grow, they make a purple color, and this is because of the oxidation of the dye. And you can quantify that when there is a growth, the more purple is the color. There is a correlation between better growth and better is the color. That means there is more growth because of the interaction, not just because by chance. And when there is white, that means there is no growth, or there is faint, that means there is a little growth. And all of these measurement was done using plate readers. So what I quantify here is we first make prediction based on monoculture with amino acid in both of the condition, and first predicted or make expectation that what happens when this intersection between these two monocultures happens. So we can say that how many carbon sources both of these monocultures utilize in common. So for instance, this interaction could be monoculture without amino acids. That means they will not grow without amino acid. In this case, the partner second will also not grow, but when we give amino acid, they will start growing. What happens when we put them in co-culture without amino acids, if they grow or not? So this means that the expectation match with the observed cases. But there are scenarios where there is a expansion, meaning none of them are growing without amino acids, only one of them are growing with amino acids, or none of them are growing with amino acid, but they can grow in co-culture without amino acid. That means this happens. So in the third case, this happens because of the interactions in the co-cultures that make the each partner growing together. Where are other cases, miscontraction can happen when both of the partner can grow with amino acids, but not without amino acid, but they will not grow without co-culture in the co-culture. This could lead to a miscontraction. So quantify this, I'll show you what I observed that. So again, the x-axis is different type of interactions, and the y-axis is number of carbon sources or number of nutrient environment they can explore when they are together. So in 41% of the cases, the predictions were matching with the observed case, indicating that the idea we have is that we can predict 50%, less than 50% of the case that there is no change in the niche. Interestingly, in 40% of the cases, we observed niche expansions, meaning that meaning that the interactions can expand the niche beyond the fundamental niche. And in out of 19% of the cases, niche was contracted. So I wondered, can phylogenetic relatedness can also explain this expansion and contraction cases, because I use different species in these settings. So I asked the distance between these species can explain the growth of expansion and contraction in these environments. So what I did again is plotted the 16 sRNA gene-based similarity, which we call phylogenetic distance, and at the y-axis I plotted number of cases of niche expansions. So what I see is a positive association, and indicating that when there is an increase in phylogenetic distance, they expand more. And furthermore, I also divided the data in a way that we can ask which type of species are doing this. Either similar species are doing more niche expansion, or between species, meaning interspecies or intraspecies interaction. So what I observed that interspecies interactions expand more niches versus intraspecies interactions. And when I asked this a similar question for niche contractions, what I observed is a negative association, indicating that more similar is the phylogenetic distance, more they are contracting the niches. And this also holds true for within species. So to summarize this part, I show that obligate cross-breeding interaction can expand the metabolic niche, and these interactions are the key driving force of the phylogenetic distance between the interacting partners can also expand the niche, and there is a positive association between this expansion and negative between this contraction. And what I showed you also is that between species interactions, or in another way, interspecies interaction actually favor this expansion. So to the fourth part, which is focusing more on the evolution part. So all the three part that I talked till now is more into the ecology, that what happens when interactions are happening in co-cultures after 24 hours or 48 hours, what is the frequencies of these interactions and what are the fitness effects? But these questions can be also asked, what happens when these interactions are happening, how evolution is acting on these interactions, and natural selection by mutation or selection is driving either interaction into productive manner or collapse the population. So to answer this, I check first that whether or not cooperative interaction evolved from bidirectional cross-breeding interaction, and then ask what mechanism actually promote these cross-breeding interactions. So this project was done in collaboration with Daniel Krusger and Lilia Musal. So to start with, I use experimental evolution of bacteria to answer this question. So where we took different bacteria, either prototrophic bacteria or which we call ancestor first, and then auxotrophic bacteria with amino acids, or they are in co-culture without amino acids, so they can exchange byproducts like bidirectional crossbreeding, not like cooperative crossbreeding. So in this case, I use only E. coli. The top one is the wild type. The middle panel is the auxotroph with amino acid, and the bottom panel is co-culture without amino acid, so they can exchange amino acid by themselves. And they were auxotrophic for tryptophan and tyrosines. So I evolved them to transfer them every 4 days, and then later decrease also to 2 days when the growth start increasing more, for total 80 days, and this means around 150 generations. The generation can be calculated based on the population size they can achieve in each transfer. So in each transfer, they were achieving around 5 to 6 generations. And what I achieve is the evolved populations that grow better. That means wild type either can grow better or worse compared to the ancestor, or the co-culture partner can either start increasing more compared to the ancestor or similar. To ask this question whether this happens or evolution actually work in these lines, I first checked the productivity of the serial transfers. So what I'm showing in the x-axis is the days of the experiment, and the y-axis the density achieved in each transfers. So what you see is that in starting the density was same, and then in the later part of the prototrophic, like the wild type, it's significantly changed. But in case of monoculture of auxotroph with amino acid, the growth didn't change that much. Then we ask what happens in case of auxotrophs when they are together without amino acids. So this is where we started with, versus when we reach in 80 days, do you see a rapid increase in the improvement of fitness, indicating that actually evolution actually increased the cooperative exchange of crossbreeding and maintain the both partners in this co-cultures. Then I ask what happens when we quantify the relative fitness of these evolved versus ancestor partners. So in evolution experiment, these experiments are really common, that people do such experiments, compare always what happens after the evolution, that the evolved population versus ancestor, do they have any signature of adaptation or selection or mutation that cannot be seen in the ancestor population, and if this happens, that you can find out phenotype that is driving this. So in our case, in our case, first we quantify that the relative fitness is compared with the ancestor. So the whatever is on the one is the ancestor population, and these box plots are showing for wild type when they are growing without amino acid, monoculture when they are growing with amino acid, and the co-cultures when they are growing together with the partner without amino acids. And you see that there is a rapid improvement in the fitness compared to the ancestor population. Then we ask what happens. Do they start producing more amino acids or not? So to test this idea, we use a biosensor that can actually quantify the production of amino acids in the environment. So what I did is with Daniel, that we paired evolved population and the ancestor populations with the biosensors. This biosensor is only there to quantify what is the increase in the production of amino acids compared to the ancestor population. So the expectation is when they are paired with evolved populations, they will grow better. The biosensor will grow better versus when they are paired with ancestor, they will go worse. So what I'm showing you is again, it's a complicated plot, but I can help you to understand. In the x-axis is the donor genotype, and the top is the recipient genotype, which is the biosensor. And what I'm showing the box plot, the one line is the ancestor, where they are compared with the evolved population. So in the wild type and monoculture with amino acid, in the tyrosine case, they don't see much different, and this holds true also for tryptophan. But in co-evolved populations, when partners are in co-culture, there is a significant increase in the amino acid production. That means evolved populations start producing costly amino acids to maintain the partners in the long run. And the letters on the top of the box plots are the significant differences, statistical differences between these. So one more interesting observation during the experiment, we observed that these partners that they are in co-culture are not only will present in single cells, but also in co-culture. So to test this, we perform a scanning electron microscopy experiment with the ancestor population and also with the evolved populations. What we observed that ancestor and evolved population for wild type and monoculture with amino acid remain single cells, but the co-cultures start showing multicellular clusters. And these multicellular clusters is actually the signature of starvation when bacteria, those are not getting amino acid, start making these aggregates. But when the partners are together, they start possibly increase the spatial proximity and then start enhancing the metabolic uptake much more better. So the production or the evolution of multicellular cluster didn't evolve in our experiment, but the cluster formation actually enhanced the spatial proximity of the partner and enhanced the exchange of metabolites in those aggregates. And this could be tested that whether or not that cluster is important in our experiment. So we performed a Nurmicocell experiment, and this device is actually is used to disrupt the interaction between two members. So this device is divided into two parts, where one part is where one partner or one bacteria is inoculated, and other part other, and they are separated either by filter or no filter. And then you can measure that the bacteria need a contact or a spatial proximity or not. This we did for ancestor and evolved population. And here I'm showing you the plot indicating that when they are filters, there is a decrease in growth in ancestor. And when there is no filter, they can grow better. But this part is less significant for evolved population, because they are adaptation during evolution experiment that can not only help them to achieve spatial proximity, but also getting amino acids from diffusion. And lastly, to crack the key puzzle, what is actually driving these interactions, we designed an experiment, and this experiment is little complicated, but I'll help you again, to understand, the experiment was done using the Nurmiko cell again. But the main question is that during the cluster formation, do they evolve any positive feedback? Because these feedbacks can increase the production and uptake of amino acid, and this can actually maintain evolution of cooperation. So what we did is we use three partner consortia, where the main partners are one partner is the producer of the and the other is the receiver. But then in this case we use a evolved strains. Then this evolved strain is actually known to produce amino acids. So but in this case we put them in 3 times lower frequency and put them either in with filter or without filter. So in this case, if this strain, the blue strain, can increase in frequency and invade the population with filter or without filter, the question is, when does this happen? So the expectation was when there is no filter, they will increase in frequency much more versus when there is filter. So and so to test this, I plotted here again the growth of the cooperator with filter and without filter, and the y-axis is the invasion success. So how better this strain, cooperative strain, can invade the population. And what I show you here is in the x-axis, you can clearly see that when they are separated by filter, there is no invasion success, indicating that bacteria, cooperative bacteria, need the partners or close interactions to invade. And this can be achieved when there is no filter, and this also holds true for tryptophan. So to indicate that bacteria in cluster actually feedback, and because of this feedback, they can maintain the populations of cooperative interactions much more versus when there is no feedback. So to summarize this part, I showed you here is that productivity actually increases in the co-culture of auxotrophs. I showed that co-culture auxotroph actually evolved costly production of amino acids because evolution actually favored the production of to maintain the co-culture population, and this they achieved by making multicellular clusters. So growth of these co-cultures are dependent on contact, and actually fitness feedbacks between these partners actually favor cooperative genotypes. So so this is all about what bacteria interact and exchange, but this is not only happen without perturbation. So I changed my focus in my postdoc. I start working on reciprocal interactions between drugs that we consume and gut bacteria that are residing in our intestine, and ask the questions what happened when they are interacting. But to start with, we all are well aware about gut microbiota in their form and function. We know that they produce several vitamins for our welfare, and then human host actually benefited for them. They help us to digest complicated plant material, cellulose or other polysaccharides. They also help us protect infections. This we call colonization resistance. So if a healthy microbiome is maintained in the gut, that the infections such as Clostridium difficile or Salmonella cannot actually flourish in the gut microbiota. But they are always something that actually affect the gut microbiota. So there are intrinsic factors also, as well as extrinsic factors that can affect the gut microbiota. So intrinsic factors can be the physiology of the gastrointestinal tract, age or host genetics, but extrinsic factors could be the diet, like more fatty diet or meat-rich diet versus vegetarian or vegan diet, or the environmental factors. But accumulated evidences also suggest that medication is the key driving force affecting the gut microbiota. So recently, there are multiple evidences that medication that are used to treat or cure or prevent diseases are directly impacting gut bacteria. This was beautifully covered by a book by Martin Blazer. So I also suggest you to read this book if you are interested to understand the basic concepts. And these evidences are not only in metagenomics-based studies, but also in vitro studies indicating that extensive impact of non-antibiotics drugs on human gut bacteria and also by antibiotics, and mostly by broad spectrum antibiotics. So in my current lab, they previously published that that non-antibiotic drugs actually impact gut bacteria. So these are not antibiotics so antibiotics are actually known to, or we used to kill bacteria either in broad ways or narrow spectrum manner. But non-antibiotics are the drugs that are not known to target commensal bacteria reside in our gut, but they are known to target host pathways, let's say antidepressant drugs or antihistamine, anti-allergic drugs, or anti-diabetic drugs. These drugs start affecting our gut microbiome, and pharmacological studies are not considering our microbiome to test these drugs and see the effects. So what the work showed that this work was done by Lisa Meyer and Naso Stippers, the supervisor in my current lab. What they did is they took 1,200 Prestwick library. These are the FDA approved drugs, and they exposed these 1,200 drugs to 40 different gut bacterial species. And these gut bacterial species are known to be obligate anaerobe. So you need a specific chamber to grow them to maintain the oxygen-free environment. And then what they did is expose these drugs and grow the growth and quantify this. And what I'm showing you here in the x-axis is the number of drugs they tested, and the y-axis 40 different most abundant and prevalent bacteria. Those are sensitized or affected by these drugs. So in the teal or green, you can see in 78% antibacterial. So they are either antibacterial or known to be antimicrobial. And in 24% of the cases, they are human targeted drugs. These human targeted drugs are known to target specific host pathways, but they find out that at least one of the drug is affecting multiple bacteria. So 24% of the tested non-antibiotic drugs actually affected commensal bacteria. What they also showed, but I'm not showing the plot, is they also demonstrated that the bacteria used very general resistant mechanism that bacteria use to actually tackle antibiotic resistance, that they also use it for non-antibiotics. That means there is a higher propensity or chances of getting antibiotic resistance when bacteria are getting resistance again non-antibiotic drugs. And the important thing to note down is that non-antibiotic drugs, we generally take it for longer, like say for years, for 5 years, 10 years. For example, anti-diabetic drug, we take it for forever, whenever we start having this diabetic symptoms. So in the start of the talk, this part, I showed that reciprocal interactions not only happen from drug to bacteria, but bacteria also changes the drug. So we know that drugs affect composition of bacteria, but there are different ways that bacteria can also change or modify the drug. So drug can inhibit the growth of bacteria, promote the growth of bacteria, but bacteria can also metabolize the drug, means decrease the efficacy of the drug, which can also affect the mode of action of the drug. And bacteria can also do the bioaccumulation. That means they accumulate the drug and decrease the efficacy of the drug. And this happens both in simple environment, laboratory environment, and communities. I'm not showing you the data, but I'm just illustrating that what I am actually investigating currently, the key questions that are interesting for me and for the lab. And for this, I'm collaborating with Camille and Nazgul in the lab So I'm testing that can bacteria evolve resistant to non-antibiotic drugs. So bacteria that can, so the drugs we take, first we expose them to good bacteria either in isolation and community, and test that can they evolve resistance against non-antibiotic drugs. And then we tested these evolved populations to different non-antibiotic drugs and asked, can this evolve resistance for non-antibiotic facilitate antibiotic interactions? So this is done like I showed previously, the evolution experiment. We have ancestor population, we evolve them in drug environment, and in this case, we increase the drug concentration, and then we have evolved population, and then we quantify or conduct experiments with antibiotics or different type of drugs to see that is there any effect in the evolved population or not. This is not only done in isolation, but also in community context. So where we have a very complex community and ask what happens when these drugs are available in community versus when they are in isolation on a single bacteria. What we are also doing is taking personalized gut bacterial communities, collecting the stool samples and in a high throughput way to quantify the effect of drugs in directly the stool samples of the patients or the healthy donors, and then seeing the changes in the frequencies of the partner, and to find out is there any emergent properties that happens in these complex communities. So to wrap up, I'm just giving you two basic implications of the work that we are doing here. Is that, so in the first part, you see that interactions are really important, not only in co-culture, but also when they are part of the community. And then when we take these drugs, they can actually perturb these interactions. And these interactions are really important because this can actually drive not only degradation of complex plants material in our gut, but also provide nutrients, and they exchange these nutrients. So if any bacteria dose is sensitive for these drugs, can be wiped out in the population and possibly never come back. And then with this, there is a destabilization of gut microbiota. And the other thing that can happen is the impact on colonization resistance. So when colonization resistance is actually in the healthy gut actually prevent the disease to happen. And when we take drugs, this can actually open up new niches for the pathogens to invade the population. And with this, you can have either dysbiosis where your gut microbiota reach completely to new stable state, or completely change from the starting part. So in this case, this can happen in 4 different ways, but the main important thing that colonization resistance provide is the resistance. So we have a resistance for bacteria to invade the population. So when the microbiome is healthy, the invader cannot actually invade the population or the resident community. But otherwise it can happen in multiple ways. So what I'm showing you here is different way to treat the data. So when we have a resident richness, like the healthy microbiome versus the invader state, in most of the cases, resistance is where invader is extinct, but in other cases, either our microbiome is destabilized or invader can maintain the populations. So just to end up, I'll recommend these books to read, to be aware about what is actually going on in grander view in terms of how microbiome shape our well-being. And to end this, I will thank all of the previous lab members, course lab members, and the current lab members in the Timpus lab, and the funding and facilities. And thank you for your attention. I'm happy to take any question.
Thank you so much for this incredibly insightful and informative talk on the ecology and evolution of bacterial interactions. I believe it is fair to say that every single audience member of this webinar will be leaving more knowledgeable and with a much greater understanding of these incredibly important yet unseen components of our environment. On behalf of the Department of Zoology, Deshbandhu College, I would like to present you with a little token of gratitude for taking the time of your day and enlightening us today. All right, sir. So it seems that the chat section is brimming with questions from the curious minds of the audience members. So with your permission, I would like to begin with a small question answer session.
Yeah, sure.
So for the first question, can the use of auxotropic recipient cells enhance the efficiency of genetic engineering techniques such as CRISPR-Cas editing?
It cannot actually enhance the. So it's an interesting question, but I never thought of it this way. But so generally, so there are two ways. So if I rephrase the question is about can we use the auxotrophs for industrial purposes or to use in genetic engineering manner. So we use a genetic tool to engineer these bacteria to study specific interactions, but these auxotrophs are also present in nature. So there are ways people do this for production of amino acids or for other purposes like vitamins, but I don't know that, or maybe I'm not aware about that. Such ways are actually already available in the. So I'm not sure actually.
All right, sir. So why does cross-feeding increase with an increasing dissimilarity to donor? What are the plausible reasons for this?
So one of the possible reason that I discuss also is that when bacteria grow, so they release a lot of metabolites in either by leakage or overflow metabolism. And this happens because the bacteria inhibition, like that, the transcriptional and translation regulation cannot directly work when there are multiple things going on simultaneously. Of course, it is fine-tuned, but still there is some leakage. And then the idea is when bacteria grow, and then when the other partners in the community are also there, they are exchanging more with this and secreting more and different type of metabolites can actually benefit the bacteria in a way that crossfeed better with the more dissimilar type of bacteria versus when they are crossfeeding with more similar types. So the pool of the metabolite they are secreting, if they are interacting with similar type, is same. So the chances are they are getting only similar types, which they can also produce other than the metabolite they are missing is one part. But when they are part of the community that are very different, they are not only getting the metabolite you need, but also other type of metabolite, which can save the cost of production of other metabolite, which can help you to increase in population size. Yeah.
Thank you, sir. So the next question would be, how do the dynamics of bacterial interactions and co-evolutions with viruses impact the potential for microbial adaptation to environmental changes?
So I didn't talk about viruses, but of course they are important, and they are also way more in number compared to bacterial populations. And there is always an arms race between bacteria and virus. I'm talking mostly about bacteriophages. Those are viruses that infect bacteria. So they are actually the key driving force of these interactions. But and there are some evidences that indicate that cross-feeding not only happen within bacteria, but also phages or bacteriophages can enhance these interactions. But I'm not sure about any specific. So this happens in all different habitats. If you talk about soil or human gut, but in what propensity, I cannot. People start actually digging deep into this, but there is not much knowledge available into this matter. Yeah.
All right, sir. So, what is the extent of commensalism of so many microbial species residing as our microbiome?
Could you please repeat the question?
What is the extent of commensalism of so many microbial species residing as our microbiome?
So it's the question is about, so in the microbiome, there are commensalistic interactions. The question is about how these commensalistic interactions affect the microbiome.
I believe so, yes.
Yeah, so commensalistic interaction is one type of interaction, but there are dynamic range of interactions, not only commensalism like mutualism, also and predation or antagonism. So these interactions happen simultaneously in one go. And because there is a spatial environment in the gut, what we study is just a snapshot of it. So even the interaction that are showing positive interaction can transition into different type of interactions when the local environment changes or ecological niches changes. So I think understanding all of the interactions are important, but for us first, breaking this complex interaction network into smaller pieces can help us first to understand how specific interaction actually play important role in different microbiomes, not only gut, but also soil microbiome or phyllosphere. But in overall, interactions are important. This is one thing, and interactions are context dependent. And I hope also commensalistic interactions are essential in at least part of life. At least in the starting, when baby are born, and then like there is a specific bacteria that actually colonize first is bifidobacterium. They are start shaping the community in the first, and they start secreting. So there's more commensalistic interaction starting, and later on these interaction changes either bidirectional. So because commensalism is more into unidirectional interactions, and this happens only when there is a secretion of metabolite or a good, but they don't want anything in return. But it's really happens, there is always a bidirectional exchange, I think.
All right. So the last question for the day, there are many questions in the chat box, but I do not wish to take much of your time. So I'll end with this question. Could it be possible to amend these methodologies in vivo conditions? If no, then what could be the possible ways to map niche specializations, independency, or microbe interactions dependencies in natural conditions? As we know, only 0.1% microbes could be cultured in the labs. Based on your expertise, you would like to know your hypothesis, assumptions, or predictions of these experiments in vivo conditions.
Is this a one question? It looks like multiple questions in one. Yeah. So I will just, I think what I understood is, of course, the question is interesting, but there are multiple layers in this. What I want to answer is these interactions are important. And I think one thing that is really important in this question is unculturability, that we know that less than 1% are culturable. And one of the key reason is because of auxotrophies, or we don't know how many different type of auxotrophies or different type of essential metabolites bacteria are not able to make their own. So we don't know what type of requirement or media requirement we have to fulfill that we can let them culture in the lab. So this is one of the key, but we can also get rid of it by growing them in a soup of communities. So we, let's say, take a supernatant of a community and give them that supernatant to let them grow. So we can increase and people are doing different ways to increase the culturability of microbiome, not only for soil microbiome, but also for gut microbiome. There are recent evidences that you can even increase this in high throughput manner to grow the bacteria. But other parts, I'm not sure how much it is.
Thank you so much for answering all the queries, sir. Now I would like to ask our co-convener of this talk, Mr. Arun Chokar, to present a vote of thanks.
Thank you, Ajita. Good evening, everyone. our esteemed invited speaker, Dr. Samir Giri, respected Principal, Sir, Professor Rajiv Agarwal, Vice Principal, Sir, Professor Kamal Kumar Gupta, Teacher in Charge, Ma'am, Professor Manisha Sengh, Event Convener, Dr. Sandhya Rai, Ma'am, and all my respected senior faculty members from our college and other colleges of university. On behalf of the Department of Zoology, Deshbandhu College, I, Arun Chokar, would like to extend my sincere gratitude to our esteemed speaker, Dr. Samir Giri, for a wonderful talk on how the bacteria are exist in communities and how the secreted metabolite of these colonies help in growth and survival of existing colony through the cross-feeding interactions. And for truly, and to Dr. Samir Giri for truly accommodating his time for his very busy schedule on a very short note. Thank you, Dr. Samir. Next, my sincere thanks to respected Principal Sir, Vice Principal Sir, Teacher in Charge Ma'am, DBT Star College Scheme Coordinator, Professor Indrakant Sir, IQSC Coordinator, Dr. Aditya Saxena, and all the senior faculty members for their support and cooperation in organizing this wonderful event. Special thanks to Dr. Monica Bajaj ma'am from Computer Science Department for helping with all the technical requirements for the talk. A wide round of applause and thanks to all the organizing team member, Sandhya ma'am, or students, organizers, Soumya, Ajita, Shubham, Aarti, Mayank, and all the participants who made the event memorable. Finally, I would like to thank all you present here for making the time to be with us today and helping us make this event successful. Thank you one and all. Now I request you all, please fill the feedback form. The link is already shared in the chat box. And I also request my senior colleagues, if anybody wants to share their views, you can share. Thank you.
We can have a photograph also, no? We can ask everyone to switch on their
Yes,
camera.
everyone, please, you can switch on your video mode.
We can have a photo, group photo. Who is taking photographs?
Ajita, please, you can take photo.
Yes, sir. Please switch on your cameras.
Thank you so much, everyone.
Thank you so
Thank
much,
you.
Dr. Samir. Thank you so much.
Welcome. Thanks for the invitation. Sudhir. Yeah. Thanks. I hope it's helpful.
It was wonderful. It was wonderful. So many questions. You can see so many questions in the chat box. Chat box is flooded with questions. And everybody is praising. So it was a wonderful talk. Thank you so
Yeah,
much.
thanks. Yeah. Let me know if something is needed and then. Yeah. Okay. Bye then.
Sure.
That's it. Yeah.
Bye. Yes. Okay.
Okay. Bye.
Thank you. Thank you.
Bye, everyone. Bye.
We can stop now.
Okay. Bye-bye, everyone.
Bye, everyone. Thank you so much, everyone. Ajita.